connecting to cloud server, and we're on.
All right. Well, so my name is Sliman Bensmaia. I'm a professor at the University of Chicago, and I am going to tell you some of the work we're doing to leverage some of our basic understanding of how sensory systems work to restore sensation to individuals who have lost it. And before I get started on the actual meat of the talk, let me just kind of talk to you about what we spend most of our time doing in my lab, which falls under the rubric neural coding. And so the idea is, I'm going to be talking about the sense of touch today. So I'm going to use that as an example. When we grasp an object, like this bottle, it causes a spatiotemporal pattern of deformation in the skin, which is what this black squiggly line is. And there are receptors in the skin that transduce this skin deformation into a pattern of neural activation in the nerve, which is what these little colored rasters are. So each row corresponds to this is a cartoon, but it corresponds to the response of one nerve fiber. And there's different colors because you have different types of nerve fibers in the skin that respond differently to the same skin deformation. So this spatial temporal pattern of skin deformation produces a spatiotemporal pattern of activation in the nerve that travels up the arm and then a short distance up the spinal cord, ultimately activating neurons in the brainstem and this structure called the cuneate nucleus. And this neural activity propagates up the neuraxis. This is a monkey brain here, ultimately giving rise to a perception of this object held in the hand. So when I hold this bottle, I can feel its shape, its size, its texture. If it's moving across my skin, I can feel that too. And somehow, all this information that is perceptually available about this object is contained in these patterns of neural activation at these various stages of processing, right? And so what we're interested in is we record from the entire neuraxis, from the nerve, from the cuneate nucleus, the thalamus, Brodmann's areas 3A, 3B, 1, 2, S2, and PV. So the entire medial limniscal dorsal column pathway. And we're interested in seeing at each stage of processing, what is it, for example, this pattern of activation in the nerve that conveys information about texture or about motion or about shape or about size. And what we do is, use these computational approaches where we can, if you give me a pattern of neural activation in the nerve, I can tell you what was the texture. If you give me two patterns, I can say, well, this was silk and this was sandpaper. I can tell you also how this texture will feel. Like, oh, this texture will feel more rougher than this other texture. Or I can say, oh, this texture is moving across the skin proximal to distally, right? So I can tell you very specific things, not only about the stimulus that cause these patterns of neural activation, but also how these patterns of neural activation will give rise to perception. That's what gets me out of bed in the morning, because what really, ultimately the biggest question for me is, how is it that nervous systems give rise to flexible, intelligent behavior? Nervous systems are better than all artificial devices in the known universe. How is it? Even like a rodent is better than any machine that exists, right? And so the question is, how is it that nervous systems are able to do that? And I like sensory neuroscience because we can control what the input is, the stimulus, where we at least can measure it. And then we can get reports about the evoked sensation, and then as we record, we can see how does the brain take this input and turn it into this output, and that I think is as good a model as any to understand what makes nervous systems be so powerful, right? That is not what I'm going to talk about today, though. What I'm going to talk about today instead is how we can leverage this basic neuroscientific understanding of, in this case, the sense of touch, to restore it to people who've lost it. And there's two populations of patients, potential users that we're talking about. One of them is amputees. So they've lost their sense of touch because they've lost their hand, right? And as I'm sure you all know by now, there is this pretty cool, rapidly developing technology of bionic hands. Like, these are robotic hands that you can control by monitoring the activity of the residual muscles, right? As you probably know, most of the muscles that control the hand are actually in the forearm. So if you have at least a distal amputation, a lot of the muscles that control the hand are still there. So you can put sensors on the muscles, and then infer from the pattern of activation in these residual muscles what the subject wants to do with the hand, and then make the robot do that, right? So the point is, I mean, this is not the topic of my talk, but the point is that these subjects can control robotic arms, or robotic hands rather, sort of by thought, right? And I'll show you some examples of that in a minute. But as I'll show you later, these hands are useless if you don't also restore the sense of touch, because when we interact with objects, we get these sensory signals back from the hand that tell us about these interactions. And so with amputees, what we can do is interface with the periphery, the peripheral nervous system. So there's a bunch of different technologies to do that. There's, intrafascicular electrodes like this one. There's extrafascicular electrodes. You can interface with the dorsal column nuclei, the dorsal root ganglia, etc. Right? And what many have shown now, and I'll show you some data later, is if you electrically stimulate the peripheral nerve or these other structures, you can evoke these very vivid sensations that are experienced on the hand, right? So that's one thing, and I'll be talking about that a little bit later. The other population of people who have lost their sense of touch are people with spinal cord injuries. So these people still have their hands, but stimulating the nerve doesn't, you can stimulate the nerve until you're blue in the face. It's not going to have any sensory experience, result in a sensory experience, because the connection between the nerve and the central nervous system has been severed. So for these people, you need to interface directly with the somatosensory, with the central nervous system. We typically interface with the somatosensory cortex, and as I'll show you, if you electrically stimulate the somatosensory cortex, so this is the part of the cortex that receives input from the skin, across the body, you can evoke these very vivid touch sensations, and that is what I'm going to start by talking about. But before I do, you might be asking yourself, why are you going to go through all this trouble to restore touch, right? And so let's talk about that for a minute. So this is a movie I made of me doing stuff with my hands. Most of those things are super trivial things, like picking things up, writing with a utensil, typing on a keyboard. These are things that we do effortlessly every day, and things that we could not do if we didn't have a sense of touch, because anytime we're interacting with anything, we get these signals from our skin that tells you about the objects, tells you about our interactions with them. And without these signals, we would struggle to do anything with our hands. Let me prove that to you by showing you a video made by my colleague Roland Johansson at Umeå University of a lady lighting a match. No problem. Later, a few minutes later, the same person is going to try to light this match. The only difference is her fingertips have been numbed with a digital anesthetic. And you can see that she suddenly struggles, she drops this match, she's like, I'm not going to be able to pick it up. And she struggles to do the same thing. And after 30 seconds struggle, she finally manages to light the match. And you can see it's abnormal the way she's doing it. And she's going to continue to struggle until the anesthetic wears off, right? Because again, she has vision, her sight, right? But the visual system is not equipped to provide the information you need to interact with objects kind of as, flexibly and dexterously as we do every day. Here's an individual who suffers from a peripheral neuropathy that has destroyed his A beta fibers. So basically, he has an intact motor system, but he has no touch or proprioceptive feedback coming from his body. And he's doing something easier than trying to light a match. He's just trying to grab this coffee cup and bring it to his mouth. And as you can see, he struggles mightily. And if I showed you this video and you had never seen it before, and you didn't know anything about this guy, and I asked you, what do you think is wrong with him? You would think he has a severe motor impairment. And this is a video that I think, as well as any video I've seen, illustrates the yin and the yang between the motor and the somatosensory system, where really one does not work without the other, right? So that's true of somebody who has an intact motor system, muscles, and everything is fine. He just has no somatosensory system. Now, here's the case we're talking about here. So this is a video from by my colleagues at the University of Pittsburgh Medical Center of a subject, Jan, you can see her head on the bottom right here, who is controlling this robotic arm by thought, right? So my colleagues at Pitt have implanted chronically implanted electrode arrays in the motor parts of her brain, the primary motor cortex. And, the idea is in the primary motor cortex, this is the part of the brain that sends signals to the muscles to move the body, right? And the idea is, even though she's paralyzed, she has a spinal cord injury. Actually, it's not an injury; it's a neurodegeneration. Even though she's paralyzed, anytime she attempts to move her body, there's a specific pattern of activation in primary motor cortex in M1 that depends on what she's trying to do. So if she's trying to move her arm in one way, you have one pattern, move her arm in a different way, you have a different pattern, right? So she can, and so my colleagues at Pitt have learned to harness these signals. So they monitor these signals and in real time infer what she's trying to do from these signals in primary motor cortex. And the models are actually very simple, and there's a cottage industry of people doing this kind of stuff, right? So if you're seeing this for the first time, which I doubt most of you are, you're like, whoa, what an amazing technological and scientific achievement that this woman can actually control this robot, this extracorporeal robot with her mind. But if you're seeing this for the 1,000th time, you'll probably notice that she's not super good at it. She's pretty slow, right? You'd be way faster than she is at this task. But the idea is, the guy I showed you just a minute ago probably would be very slow at this task too. And so the idea is that if you want to create a dexterous bionic hand, you need not only to have a sweet robot and ways to read out intended movements from the brain, but you also need to restore the sensory feedback. And that's what we're setting off to do, right? So how are we going to go about this? Well, as I said earlier, anytime you touch something, the skin deforms in a way that depends on what you touch and how you touch it. You have these receptors in your skin that transduce this deformation, and these neural signals about this interaction travel up the neural axis, ultimately giving rise to a pattern of neural activity in somatosensory cortex. So this is the part of the brain that receives input from the body. And that pattern depends on what you touch and how you touch it, right? So in these bionic hands, there are sensors. And what we're working on is taking in the output of these sensors and converting them into pattern of electrical stimulation of the brain to reproduce, to mimic to the extent possible, these natural patterns of activation, right? And if we could completely mimic natural patterns of activation in the brain, we could produce completely natural sensations, like restore your ability to tell cheap silk from good silk. We are not there yet at all, but as I hope to show you, we're getting there. We're moving toward that. So let's talk a little bit about the somatosensory cortex. This is where we're going to put our arrays. As all of you know, I'm sure, there is the somatosensory homunculus. There's a body map in somatosensory cortex. That means that neurons near the midline respond to touch on the bottom, the bottom half of the body. Neurons more laterally respond to the hand and the face. And even within the hand representation, there's a very clean separation between thumb, index, middle ring, and pinky. And so we're going to put our electrodes in the hand part of the brain, right? And the way you find the hand part, the hand representation in paralyzed, insensate subjects, is that you have them move, imagine moving their fingers, and that lights up when they, imagine moving their thumb, that lights up the thumb representation in somatosensory cortex, and then you can read that signal using an fMRI or a MEG, right? Which is MEG is what's shown here. So, right, so we're sticking our electrodes in the hand representations in somatosensory cortex. Now, I started out this whole spiel talking about how we want to leverage what we know about how the brain encodes sensory signals to try to restore the sensory signals, right? And I told you there's a body map. I mean, this is like an old this has been known for a long time, that there's a map of the body in somatosensory cortex. And now let me prove it to you, it's been, again, shown a bunch of times, but this is the pattern of neural activity. I forgot to mention this here. So here, the kinds of arrays that we're implanting in somatosensory cortex are called Utah electrode arrays. These are the only arrays basically in the world that you can implant in a human that penetrate into the somatosensory or into the cortex, right? So these are penetrating electrodes. This is a 4 by 4 millimeter area. There's 100 electrodes, so the pitch between adjacent electrodes is 400 microns. So this is the pattern of neural activity over a 4x4 millimeter area, right? This is recorded from an array like this. This is in a rhesus macaque, right? And what we did here is we poked the rhesus macaque in the second palmar whorl, and when we do that, we get a pattern of neural activation that peaks right over here, right? So red means very active, blue means not active at all. Intermediate colors mean intermediate levels of activity, right? So when we poke the animal here, we get a pattern of activity neural activity that peaks here. When we poke in a different spot on the palm, the pattern of neural activity shifts to a different part of the brain. You poke the pinky, you get a different pattern of activity. So this is what I just told you earlier. I'm showing you that there's a map of the hand in the somatosensory cortex. And what we're going to try to do is leverage this map. So the idea being, if I electrically activate neurons that respond to the index finger, can I evoke a sensation on the index finger? So that's what we're going to try to do. I did a bunch of experiments in monkeys that led to the following experiment in humans. So the idea here, what I'm going to show you is a video where there's sensors, like I told you, there's sensors on the hand, where the sensor on the index finger, on the bionic index finger, is connected to electrodes that respond to the index finger, right? The middle finger on the sensor on the bionic middle finger is attached to the middle finger representations, etc. And so what I'm going to show you is Rob, my colleague at Pitt, is touching different fingers in random order and asking Nathan, who's the subject, which finger did I touch, right? And Nathan is simply reporting where he feels it on his own hand, right? So he doesn't have to learn what these sensations mean. He's just going to say, oh, I felt something on my index. Oh, I felt something on my ring finger, whatever.
I'm good.
Okay, here we go again.
Index. Ring.
So spoiler alert, he's going to get them all right. And, so as you can see, Nathan has two pedestals coming out of his head here.
Middle.
One of them, because he has two motor arrays and two sensory arrays. So not only can he move a robotic hand now, but he can actually feel through it. And I'll show you some more videos in a minute. But this shows you that we can create, when you're grasping an object, you need to know.
Index.
What do you need to know? You need to know that you're touching it, but also where you're touching it. Minimally, the thumb and your fingers needs to be touching the object for you to be able to grasp it. And the way to convey information about which part of your hand is grasping the object, you can do this somatotopic mapping where you link sensors on the hand, on the bionic hand, to the corresponding representation. So the thumb representation, the index representation. And when you do that, it's very intuitive of where the bionic hand is touching the object. So the second thing you need to know to convey is how much pressure you're exerting on an object. We are exquisitely good at exerting enough pressure on an object so as to not drop it, but not so much as we're squeezing it unnecessarily, right? We just apply enough pressure on objects. And the way we do that is through our sense of touch. So if I, for instance, anesthetize your hand, you're just going to be either dropping objects or jamming your hand into them because you have no idea, you have only very little idea from your motor system and your proprioceptive system how much force you're exerting. Most of that really fine information about how much force you're exerting on objects comes from the sense of touch, right? So then the question is, how is it that the nervous system encodes that information about force, about the force that we exert on objects, right? And so we looked at our monkey responses. So again, this is the 4x4 millimeter area in the somatosensory cortex of a rhesus macaque. And here we're poking the pinky at three different levels of pressure. Low pressure, so a faint touch on the hand, which creates a faint sensation, and then high pressure, which is, we jam, well, it's not really that high, but we press pretty hard on the skin, and this is the pattern neural activity, right? And this is an intermediate level of force. And as you can see, two things happen when you increase the pressure on the skin. One, this little, the neurons here in the most active part become more and more active, right? And two, more and more neurons, you can see that area of activation increase. More and more neurons become activated by the stimulus, by this pressure stimulus. So that's what we're going to try to do, right? That's how the brain encodes pressure. So we're going to try to reproduce that through electrical stimulation, right? And the nice thing is there's an easy way to do that, and that is just by cranking up the juice. And by juice, I mean electrical current. The idea is if you inject low current, you're going to weakly activate neurons near the tip of the electrode. And as you increase the current, you're going to more strongly activate neurons near the electrode, and it's going to spread further out, right? And so that's kind of, at least qualitatively, similar to what happens when you increase the pressure, right? So we did, again, a long series of experiments, to understand how much do we need to electrically stimulate the brain to create an appropriate level of pressure, right? And I'm not going to bore you with this interminable series of experiments. I'll just tell you the following thing, which is we came up with a mapping of electrical amplitude on pressure, which basically tells you if you want to create a sensation that is equivalent to, let's say, a 500 micron, this is microns of indentation into the skin. I'm just calling pressure for ease of communication. 500 microns of indentation into the skin, you need to stimulate the brain at 65 microamps, right? And if you stimulate the brain at 65 microamps, it's going to feel as strong as if you indented the skin at 500 microns. So we came up, I haven't explained to you how we got this function, because it would take, you know, the entire talk to do that. But what I'm going to do, and I should say these two different colors correspond to two different monkeys. One monkey was super consistent, the other monkey was less so, but you can see it's all these curves basically have the same shape, right? You have this sort of negatively accelerating function of amplitude over pressure. But what I will show you is that this function works by showing you how we deployed it in a monkey experiment. So what we did is we had trained these animals to discriminate pressure. So we would poke the animal twice in the same location. One of the pokes was stronger, and then the animal just learned to report which of the two pokes was stronger, right? And obviously the relative amplitudes varied or whatever. Once the animals were trained to do that, what we did is we had the animal do the same task, but this time, instead of poking its skin, we poked a prosthetic hand. And then in real time, so this is the output of, this is one example trial. This is the output of the pressure sensor on the prosthetic hand. In real time, we converted this pressure output into electrical stimulation based on this function I showed you earlier. And that's what these rasters are here, right? So this is the electrical pulse trains as a function of time for these two intervals. And the animal has to do this pressure discrimination task on the basis of this artificial tactile feedback about pressure, right? And here are the results. So if you do this task and on the native skin, on the hand of the monkeys are intact, right? The purple trace shows you how well the monkey do. So the monkeys do. So basically, as the difference in pressure increases between the two tasks, the two intervals, they become better and better at reporting which of the two contain the stronger stimulus, right? So that's what the purple trace shows. The two magenta traces show how the animals performed on the same task, but the information they get about the pressure is through this prosthesis, right? And you can see they're just as good. So they're just as good at discriminating different levels of pressure, whether that pressure is applied directly to the skin or whether it's applied to a prosthetic finger and they get the information via this direct interface with the brain. So this just basically tells you that this function does what I told you it did, which is map electrical amplitude onto pressure, right? So now we have a objective or sort of rigorous way to take the output of sensors and convert them into patterns of electrical stimulation to produce sensations that are appropriate to the level of pressure that was exerted, right? So how do we put these two together? How do we put these two things together? So here in the next, what we did here, and again, this is where at Pitt, is we took these two concepts that I told you about. The first concept being you take the thumb sensor, attach it to the thumb representation, index sensor, index representation, and then the second one is you basically modulate the amplitude of the stimulation depending on the output of the sensor, right, on the hand. And when you do that, so this is Nathan now, and he's doing this task that I showed you Jan was doing earlier. But again, on the left, though, he can feel the object when he makes contact with it through this brain interface, right? And on the right, they turned off the stimulation, right? And you can eyeball this and see, and so these are, on the left and the right, he's grasping the same objects with and without this artificial touch, right? And you can see from this video, and this is published in Science last year, if you want to see the actual data, that he is much faster doing this task with stimulation than without, right? So that's basically the state of the art right now. And the state of the art is what I told you, like you have, if this is like the initiation, maintenance, and termination of contact with an object, the way the stimulation is going to go is you're going to increase it during contact initiation, you're going to maintain it during contact maintenance, and then you're going to terminate it when the contact is terminated, right? But when you look into the brain, that is not how the brain responds, right? In fact, if you indent the skin, hold the indentation, and then you terminate the indentation, you have these huge transients at the onset and offset of the indentation of this, contact of this force. But during maintained contact, you can see the neural response dwindles to almost nothing, right? So there's something missing in the way we're conveying sensory feedback right now in this kind of standard way, in that it doesn't capture these dynamics, because it turns out that the nervous system, the sense of touch, this is throughout from the nerve through the entire neuroaxis, really responds much more to changes in contact than it does to maintain contact, right? So it really emphasizes these contact transients, onset and offset, over these kind of where, contact is stable, right? So the next generation of prosthetic hands needs to build this into the algorithms, right? And this is where I'm going to pivot and talk a little bit about amputees, because this concept has actually been implemented in amputees and is only now beginning to be implemented in humans. And I'll show you some preliminary data on that later. But so this is a video from my colleague Dustin Tyler at Case Western University, really one of the world leaders in restoring, hand function through these bionic hands. And one of his, he's been one of the major pioneers in the restoration of touch there. I showed you there's a bunch of technologies. His technology specifically is an extravesicular electrode. So he electrically stimulates the nerves, the residual nerves. And so let's think a little bit now. Let's take a step back and say, what if we wanted to restore touch in all of its glory? Restore the ability to sense texture and shape and motion, all of that. What would you need to do? Well, you have something like 18,000 nerve fibers that innervate the palmar surface of your hand, right? Anytime you interact with an object, all 18,000 of these, a large number of these 18,000 nerve fibers is activated, and almost every one of them is conveying a different message to the brain. And that message depends on what kind of nerve fibers it is. I told you there are different types of receptors that respond differently to mechanical deformations of the skin. It also depends on where the receptor is on the hand, right? So for instance, if you're making contact with your index and your thumb, index and thumb fibers are going to be much more active than pinky fibers, right? And there's idiosyncratic differences across fibers, and there's all kinds of complexity there. If you wanted to restore touch, what would you need to do? Well, you would need to be able to activate each one of these 18,000 nerve fibers independently of all the others, selectively activate it. And you would need to know, how would this particular nerve fiber respond to this particular, to whatever the bionic hand is doing right now, right? So you would need this massive, massive stimulator that allows you to activate these 18,000 nerve fibers independently, and you would need to know how to activate them. And while the technology is not there, the science basically is. So I'm not going to go into details. This was published like 4 or 5 years ago in PNAS, but What we've done is, and this is, sort of culminated in the work of Hannes Saul and Benoit Dallet, is to create a model of the nerve. And basically how this model works, of each, it's a model that predicts how each of the 18,000 nerve fibers in the hand would respond to whatever stimulus you can come up with, right? So what you do is you specify what the pattern of skin deformation is. And what the model does is first, estimate how the skin would deform, because a lot of our sense of touch is actually determined by the mechanical properties of the skin. So the first thing this model does is figure out how would the skin respond to this stimulus that you're applying to the skin. And then it tiles the skin with, afferents at their known densities, because there's different, you have many more afferents that innervate the distal part, the fingertips, than other parts of the hand. It tiles the hand with these afferents at their known densities, and then it simulates how each of these afferents would respond to whatever the local skin deformation is, right? Anyways, the details of the model don't matter for the purpose of this talk. The point is, though, that this model just spits out the response of all, well, in this case, it's 12,000 nerve fibers, because this is built on a monkey, but monkeys and humans have basically identical innervation of the skin. So it spits out how each of these nerve fibers would respond to whatever stimulus you can come up with, right? So so this model, again, the point of this PNAS paper is to show how this relatively simple model can really reproduce all the known, or most of the known, response properties of nerve fibers. So for instance, not only can it reproduce the firing rates, the strength of the response of different nerve fibers to a bunch of different stimuli, but it can also reproduce the timing. So if you deliver a stimulus to the skin, each of these nerve fibers is going to produce a very, very temporally precise response to it, and the model can reproduce that temporally precise response. So it can, capture really all this complexity in the nerve response. This is just to show you how the model looks, what it looks like. So this is here, the user here for the models specifies an edge indented to the skin. This surface shows you how the skin would deform in response to that edge. And then this is just a response of the fingertip to two edges at two different orientations for the three different nerve fiber types. And for instance, you can see that SA1 fibers respond throughout the indentation. RAs and PCs only responds at the onset and offset. Anyways, this is just a kind of some eye candy to give you a sense. Every time something twinkles, it means an afferent got excited. It's just to give you a sense of how this model works. So this model reproduces the neural activity of all 12,000 nerve fibers, right? So if we had a stimulator that allowed you to selectively activate thousands and thousands of different nerve fibers individually, then that would be, we could restore the sense of touch and we'd be good to go. Problem is, technology, that's not where technology is right now. Where technology is right now is you can activate hundreds of afferents at a given time, or maybe even thousands, right? So there's no point in having a model that can tell you what each nerve fiber is doing if all you're doing is activating hundreds of them at a time, right? So what you need to know is how would hundreds of nerve fibers be activated giving the stimulus, right? So my grad students, Lisa and Shin Pu, create a simplified model, which basically reproduces with many fewer parameters. That this model is like a trivially simple model, how aggregates of nerve fibers would respond to a stimulus, right? So this is just to show you, again, this is published, so I'm going to kind of fly through this. I just want to give you a sense of this. So this is like, we did an experiment a long time ago, so at Hopkins, where we just measured the pressure. We had a sensorized glove, and we just monitored the pressure as subjects perform a bunch of activities of daily living, like opening a door, using a mouse or a keyboard or whatever. These color traces here are the forces that were measured with this sensorized glove. The black traces here show how it's the red traces show how the nerve would respond. This is actually simulated with the full model, how an aggregate of afferents would respond to the stimulus. And then the colored traces show how this much simpler model can reproduce that aggregate activity. So the point there's 2 points I want you to see from here. One, when you're looking at aggregates, you can predict them, how they would respond to these stimuli with many fewer parameters. So that's the main point. The other point, though, that's also important is to show that, again, like I showed you earlier, these are the force traces, right? If you use the standard sensory feedback approach, you are going to be tracking these force traces as they are. These are the neural responses to those force traces. They look nothing like the force traces, right? But that's how the nerve would respond. So what we're going to try to do is say, this is how the nerve responds. Let's do that. Let's try to stimulate the nerve to reproduce these patterns of neural activation at the aggregate level, which is the only, which is the best we can do with the current technology. And when we do that, and by we, I mean colleagues of mine in various places, I'm going to introduce you in a second, good things happen. So this is work from Silvestro Machera's lab at EPFL, showing that when you have these biomimetic trains, right, you can evoke more natural sensations, right? If you try to reproduce how the nerve works, you get these more natural sensations. Go ahead, do you have a question? Anyone?
Oh no, it was just the clap emoji.
Oh, I see, cool.
Yeah.
Sorry, my bad. I thought there was a raised hand. So you get a more natural sensation. So that's promising, right? Not only that, but you can actually, subjects can use their hand, the bionic hand, more effectively. So this is a task where the subject had to transport a fragile object across this barrier, right? So if she squeezed the object too hard, it broke. I mean, it didn't really break, but it was like it fake broke. And so, the test was, how many times can you transport this fragile object across across this barrier? And she could transport more of those objects without breaking them with the biomimetic feedback than without. And right, so this is what this shows. And then another colleague of mine, Greg Clark at the University of Utah, was able to reproduce that task that I just showed you, but did another one where he had subjects discriminate the compliance of objects. Obviously, they couldn't see the object, and they had to squeeze the object with his bionic hand. And so they had to identify various levels of compliance of the object with the standard feedback and with the biomimetic feedback. And this was too easy, so the subjects were really, really good at this task, right? They were nearly 100% because these objects were too similar to too different from each other. But they were much faster with the biomimetic feedback at doing this task than they were with the standard feedback, right? So it feels more natural. It allows you to better manipulate objects, and it allows you to better sense the properties of objects grasped. So that's, so that shows, I feel like now there's a mounting evidence that this biomimetics, sort of this mimicking of natural patterns of neural activity in their dynamics, produce these more natural and more intuitive sensations. So, but now let's circle back to the brain, right? So this is part of the same study I showed you earlier, and I showed you when you press on the skin and then release, right? You have these transients at the onset and offset of contact. And then during maintained contact, the activity is really weak, right? So this is true in the nerve, and this is also true in the brain. So at the aggregate level, cortical neurons are very, very different from afferents individually. But when you look at 100 cortical neurons and 100 afferents, they look similar, right? So the idea is these principles that we've applied successfully to the nerve, this biomimetic sensory encoding algorithm that we've applied successfully to the nerve, we can take this exact algorithm and port it to the brain, and it should work, right? And I'll tell you what, none of what I'm about to show you is published, but it's looking pretty good. So this is, again, my colleagues at Pitt, they just simply had their subject, Nathan, they presented him with a stimulus that was either fixed, like flat train, like a standard flat train, or that was actually replaying the neural activity in the brain, right? And basically they found that when you replay the neural activity in the brain, the subjects found that much more natural than when they played these fixed trains, right? These fixed trains, these flat trains, do not mimic the way the brain normally responds. And therefore, when you activate the brain using these fixed trains, you get these unnatural percepts. This is like Scott, we have, recently, that was 3 years ago, started doing experiments in my lab with a human subject, Scott, who's just the best. And we were able to reproduce that. On most electrodes, he felt these biomimetic trains to be more natural than the fixed ones. This was if you pair biomimetic and fixed at equal magnitude, he chose the biomimetic one on 6 of 9 electrodes with a lot, with a strong preference. Right, so I'll mention a couple other things. A couple of things that are actually going to come out in the next couple of weeks. It turns out that biomimetic feedback has other advantages. So it's more natural, more intuitive, and supports better dexterous movements. It allows you to give me more information about objects. But also what we've discovered recently for brain-computer interfaces is that when you electrically stimulate sensory cortex to convey sensory feedback, you get some activity evoked in M1. And that activity that you're electrically evoking in M1, in motor cortex, actually interferes with your ability to read out the intended movement from M1, right? But if you use biomimetic feedback, you only have these brief transients at the onset of contact, much weaker stimulation during maintained contact, so that minimizes that contamination effect. The other thing we discovered that I'm very excited about is that when you grasp an object 2 times, 1, 2, just let's say you're like grasping 2 objects and you're trying to see which of these is more compliant. And if you grasp them within 5 seconds of each other, and you're using kind of the standard approach where you're titrating the amplitude according to the force applied, what happens is the second object is going to feel, it's going to feel like you're applying more force on the second object than the first, even if you're applying the same amount of force. Because somehow these flat trains send the brain into some kind of different state, and it takes seconds to kind of get back to baseline, right? But a biomimetic train doesn't have that effect because it is so brief that it does not kind of send the brain into this different state, and therefore gives rise to much more stable sensations, right? So, and then the other thing that's maybe obvious is that you need less power. You need less power, electrical power, less battery consumption with these biomimetic trains, because you don't need to maintain these high levels of stimulation for an extended period of time. So basically, the main point I want to get across is that mimicking how the nervous system encodes information in these artificial sensory encoding algorithms has a bunch of advantages and is definitely the way to go. I'm going to end by saying that this is a major international effort. I have colleagues all over the world, trying to restore sensory motor function in people is hard. It takes a village, and so these are all my colleagues working. This is just my prosthetics colleagues throughout the world, and this is my lab. These are my funding sources, and yeah, thank you for your attention. I guess I have time for a few questions if there are any.
Wow, thank you so much for this talk. Honestly, there is so much out there that I did not know about. Basically what's currently going on in the research field. But I guess just to save time, we'll wait if there's any questions. And because I have a few myself, but let's see if there's any from the audience. I think if there aren't any, then okay. So yeah, so this reminded me of this episode I saw on one of these shows that I like to binge watch, basically. It's called The Good Doctor, and they have an episode where they gave a patient a whole new wrist. The hand kind of looked similar to one of the hands in the videos that you showed. And so I was curious how basically these prosthetics, there is no, let's say, immune response from the body. Like, I'm curious how that works. How do you deal with the fact that, the body might reject the prosthetics? But especially seeing that it's the hand, a very sensitive part. So I just wanted to see if you had any input on that front.
Right. I mean, that's not really what I do, but I think these prostheses, there's the main approach to bionic hands is just to kind of like, there's a socket, right? And so the hand does not need to be biologically integrated with the body, right? The only invasive part is the wires, hopefully one day it'll be wireless, but right now it's still wired, where you have to have some kind of percutaneous connection with the nerve or with the brain to be for the bionic hand to be able to communicate with it with the nervous system, right? But the hand itself is not really interacting with the inside of your body. It's only interacting with your skin.
Oh, I see. And when you say wireless, do you mean that maybe one day people with these additions per se will be able to, let's say, control whatever extra body part they have via, let's say, an app, or let's say something on their mobile phone?
Yeah,
Is that
basically,
something?
right. So the idea now, right? Everything I showed you, you could see wires coming out of the head, right?
Yeah.
With the amputees, it's not clear, but there are wires connecting the sensors to the nerve, right? But there's technology already exists. It just hasn't been approved in humans yet, but it exists it's used in animal experiments where you can harness the sensor output and electrically stimulate, through entirely implanted arrays, right? That are inductively charged with an external charger. And so that technology exists, right? It's, because this hasn't been deployed in the field yet, it hasn't really been, there's no need for it yet. But as soon as it's, before it's deployed in the clinic, they will, that wireless technology will be deployed, right?
Yeah, I suppose it has to go through the clinical trials and whatnot, right?
Yeah, and we're doing preclinical trials right now. But the broader question that I think I heard you ask is that right now we're working on restoration, right? We're working on people who have lost their hand or people who have lost their ability to control their hand, right? But ultimately, these kinds of technologies can be applied in a variety of other ways, for good and evil. But, like, for instance, Elon Musk's company, Neuralink, right? The idea there.
I was about to mention that. Yeah, I was about to ask about his company. And the whole idea of biohacking. I'm wondering what your opinion is on the field.
Right. So let's start with Elon Musk thing, right? So his idea is, he's not so interested in restoration. I mean, he gives it lip service, but ultimately what his goal is to create these interfaces where you can harness signals from the brain to control extracorporeal devices of any sort, right? Mm-hmm. So you can control your computer just by thought. You can control your car just by thought, whatever. You can communicate with other people just by thought, right? And so that involves the same kind of ideas where you're trying to make sense of signals from the brain, right? Here, we're just trying to control alarm, but you can do other things with it. And you can pump signals back into the brain. Here we're trying to restore the sense of touch, but you can do other things. You can restore vision. But ultimately, imagine if you could sort of pump information to the brain at a much higher bandwidth, so you could have access to the whole of human knowledge, like via Wikipedia or something. If you could interact with Wikipedia the way you interact with your own semantic and episodic memory, right? So those are the kinds of
med school wouldn't take 6 years.
Was that?
I was saying that med school wouldn't take 6 years.
You wouldn't even need to go to school. You wouldn't need to go to school. There it is. It's already there. So, I mean, it's not going to happen in our lifetime, probably.
Probably not. No.
But, it's a thing that maybe eventually could happen. We'll see.
I mean, from what I've seen so far, there's this community of biohackers. Well, mainly, teenagers, people in their early 20s, doing all sorts of things to give themselves functionalities that nobody else has, but nothing that actively serves a purpose in their life, nothing that actually has an end goal to it. Maybe something like having these lights on their wrists, or having these, just some, So,
well, I don't think that you can't biohack your nervous system and live to tell the tale. I mean, that's a terrible, terrible idea. So I don't think but you know what your question about biohacking did bring up is this idea of privacy, right? Imagine we're in the future and you have these implants in your brain that really kind of have access to all your innermost thoughts, right? There are people working on, from brain scans, can I infer what this person is thinking? And the answer is not completely, but a little bit. And as technology improves, that ability will improve, right? And it's going to, as these devices have more and more access to your innermost thoughts, it is going to be more and more important to make sure that no one can have access to those, right? And so that's people actually have those conversations and try to figure it out, try to make sure that there's enough of a security protocol that your thoughts remain your own, if that's what you want.
Mm. And I think that also goes under the whole, from, right now in this domain, or rather like in a broader domain per se, you see all these laws being put into action with regards to artificial intelligence in medicine. So if an AI system messes up, who's held responsible? The doctors, the patient, maybe because the patient agreed to something that wasn't exactly 100% thought out, and so on and so forth. So I think that, at least from what I've seen, we're probably right at the beginning of the road. There's still so much work to be done, or at least that's the impression I'm getting.
It's a really good, the analogy of AI kind of in medicine is, so there's now, it's obviously great, AI is really good at certain things that humans are not, especially when they have access to a lot of information, right? Which you do in the medical field, where hospitals keep very detailed record of everything, right? So I think there's a lot of promise there, but there's also some ethical considerations that you start to wrestle with. Well, in brain-computer interfaces, there is another slew. It's a different slew of ethical considerations. Different, right? But I assure you that people are thinking about those. And when we have these meetings, there are ethicists there are philosophers there to think, hey, wait a minute, let's think about these other things. Okay, we have a question here. Is there a way to restore the sense of sight, for example, in a way? Oh, yeah, absolutely. Those exact same principles that I talked about today for touch are being applied for vision, right? So there's two different types of approaches for visual restoration. One of them is the retinal implants, right? So you have receptors in your eye that transform photons into neural signals, and so you can electrically or even optically activate these neurons to restore vision. Or you can even use the brain-computer interface route. There's the primary visual cortex is in the back of the brain. You put stimulating electrodes in there and the same things apply. But I am much more skeptical of visual neuroprostheses than I am of tactile ones. Not only because I'm doing the touch, but if you think about what you need for a vision, right? Our visual, there's millions of photoreceptors in our eyes, as opposed to thousands of receptors in our skin. And to get to perceive an object, you need a lot of receptors to be activated simultaneously and in ways that make sense, right? I think the challenge is much greater to be able to restore vision, at least object perception and vision is going to be a huge challenge. With touch, as long as I can even tell you that the thumb and the middle finger are touching, that is better than nothing. That's going to get you a fairly long way. Just having two phosphenes that can fire independently, that is not going to get you very far. So I think while the same principles apply to vision, I think the challenge is much greater. And they've been working on retinal implants, and those things are still not, in clinical. I mean, there's some clinical flirtations, but not all the companies that tried to do that have gone out of business. So it's I think a longer term project. And on that note, I really should go because I have to be in surgery soon.
Well,
But
thank you
thank you
so
for the
much for your
invitation, and
time.
thank you for your
Yeah,
attention.
this was quite interesting and great to watch. I'll make sure that the recording is up and ready, and Dr. Neil will upload it onto YouTube later on. So
Awesome.
I'll stop the recording right here.